Hey guys, welcome to the new video. Today we'll go over the lecture portion of the exam. This is the reproductive system, and yeah, let's just get over it. So let's begin. This is the female part, by the way. So what oogenesis basically means is the development of an egg cell. Where that happens is in the female's ovary. So for an egg cell to develop, two events must occur, which is the meiotic event and the follicle development. Instead of me teaching you, I will play the video where I learned it from because I kind of struggle to explain it, and I think it's better for me to play the video where I learned it from, so you guys can learn it too. And so yeah, I'll play the first video here. This video right here is the follicle development, and then the next one I'm going to go over is the meiotic event.
There are two processes involved in the development of an egg cell. There's follicular development. That's what this video is about. It's about the cellular package that contains the oocyte. And then there's also the meiotic events that give rise to the oocyte itself. So just quickly, the highlights of the meiotic events are that meiosis begins prior to the person being born. And so that's why the population of oogonia in the ovaries are known as primary oocytes, because they've entered meiosis. But then they stay inactive. They stay like that until puberty. And so they're quiet until puberty. And then starting at puberty, some of the primary oocytes present in the ovary will continue meiosis where they left off. And that first meiotic division that gives rise to the secondary oocyte and the first polar body occurs just prior to ovulation. And so ovulation is the release of the secondary oocyte. Notice that at puberty, when some of the primary oocytes start to continue in meiosis, that the cellular package, or in other words, the follicle, will also start growing and getting bigger and more complex. So let's take a closer look at the changes that the follicle goes through. In this diagram, we can see the type of follicle that a female is born with. A female is born with many small follicles that kind of cluster together on the outer periphery of the ovary. They're called primordial follicles. In the micrograph, we can see some primordial follicles, and these primordial follicles consist of a primary oocyte with a single layer of squamous cells, flat cells, called follicular cells. Once the person enters puberty, hormones from the brain, particularly follicle-stimulating hormone, or FSH, good name, is going to cause some of these follicles to wake up. It's going to activate them. And when the follicles first start responding to hormonal changes, the primordial follicle is going to differentiate into a primary follicle. Taking a look at the primary follicle, the oocyte is still a primary oocyte, but notice how the follicular cells are no longer squamous. They're now more cuboidal or columnar in shape. And so the primary follicle is much larger than the primordial follicle. What happens next is the follicular cells start to divide by mitosis, giving us multiple layers of cells. This is what it looks like. Once you have more than one layer of follicular cells, as we can see here, there are multiple layers. Those cells are now known as granulosa cells. And once we have multiple layers of granulosa cells that surround, this is still the primary oocyte, then the whole structure is called a secondary follicle. In a secondary follicle, the primary oocyte starts to deposit a layer of sugar and protein on the outer membrane of the oocyte, known as the zona pellucida. And then the granulosa cells that are growing in number also have a few jobs. One thing that they do is they produce and secrete follicular fluid, which starts to accumulate within the follicle. Granulosa cells also produce estrogen. And so as the follicle is growing bigger in the ovary, estrogen levels are going up. Taking a look at this follicle, this is still a secondary follicle. We can see the outer periphery of the follicle here. And we can see that all those pink cells are granulosa cells. Notice the pools of follicular fluid that are starting to accumulate within this secondary follicle. This is still a primary oocyte because remember, the first meiotic division doesn't occur until just before ovulation. We can also see the zona pellucida surrounding the oocyte. Another thing that we can really notice here is that the ovarian tissue that's surrounding the follicle, this region, is starting to change to support the life of the follicle. That ovarian tissue surrounding the follicle is known as the theca folliculi. It consists of two layers, the theca interna, which is a rich blood supply to the follicle, and then theca externa, which is a connective tissue encasement of the follicle. Eventually, as the secondary follicle grows bigger and accumulates more follicular fluid, these little pools of follicular fluid are going to merge into one big lake, one big cavity of fluid known as the antrum. Once the follicular fluid merges into one big cavity called the antrum, we can see that here, this follicle is known as a vesicular follicle, also known as a tertiary follicle or a Graafian follicle. We can see all of the main parts that we talked about. Here's the outer periphery of the follicle and all of the granulosa cells below that in dark pink, still secreting estrogen. We can see that there are granulosa cells that are kind of separating the oocyte up on a stalk, and granulosa cells surround the oocyte. Those granulosa cells are known as the corona radiata, and they're surrounding the zona pellucida. And of course, the cell is still a primary oocyte until just before ovulation. What will happen to the vesicular follicle is it will start to bulge from the surface of the ovary. Taking a look at this diagram, here's the vesicular follicle, and this is what I was trying to describe. It will actually start to bulge from the surface of the ovary, and just prior to ovulation, that's when the first meiotic division is going to occur to give rise to the secondary oocyte, which is the cell that gets ovulated, and then the first polar body. Here's a photograph of the ovary just prior to ovulation, where we can see the ovary with the whitish appearance in the background, and we can see the follicle bulging from the surface, and then you can even see the oocyte about to be released. And so you can see it's like a blister that forms on the surface of the ovary. After ovulation occurs, the empty follicle repairs itself, and it actually thrives and becomes a structure called the corpus luteum. The follicle that no longer contains the oocyte is still secreting hormones too. We know that the growing follicle containing the oocyte secretes estrogen in increasing amounts as it gets larger. Once ovulation occurs, estrogen levels drop because that follicle is damaged. But once the follicle repairs and becomes a corpus luteum, those cells primarily secrete progesterone, and so progesterone levels start going up. The corpus luteum has a limited lifespan. Eventually, it starts to die off, and that's known as the corpus albicans. Here we can see the microscopic appearance of the corpus luteum, and just to compare that to the corpus albicans, you can see how it looks like the same structure, but it's shriveled up.
All right, going over the meiotic events, I'll play that right now.
In this lesson, we'll explore the details of what happens during the phases of meiosis. Meiosis, sometimes called reduction division, is the type of cell division that produces gametes. By gametes, we mean sex cells, such as sperm cells in males and egg cells in females. Meiosis is broken down into two stages of cell division, called meiosis 1 and meiosis 2. Meiosis 1 has 4 phases: prophase 1, metaphase 1, anaphase 1, and telophase 1. And meiosis 2 also has 4 phases: prophase 2, metaphase 2, anaphase 2, and telophase 2. Let's look at what happens during meiosis 1. Prophase 1 starts with a diploid cell. Its chromatin contains 2 uncoiled, spread-out sets of chromosomes, 1 from each parent. After the DNA in the chromatin replicates, it condenses into the more familiar X-shaped chromosomes. The replicated DNA is the same in the identical sister chromatids of each chromosome. In a process called synapsis, each chromosome pairs up with and binds to its corresponding homologous chromosome, forming a tetrad. A tetrad is the group of 4 sister chromatids in paired homologous chromosomes. The chromosomes contain genetic information called genes. These genes were inherited from each parent. And different versions of the same gene on each chromosome are called alleles. In a process called crossing over, chromatids from each homologous chromosome exchange segments of alleles. Also called recombination, crossing over randomly happens on every chromosome, resulting in different gene combinations. This explains why every gamete is genetically different from every other gamete. Crossing over results in genetic variety in offspring. This is why children are different from their biological parents, as well as from their biological siblings. Continuing on with prophase 1, the nuclear membrane disappears. The centrioles move to opposite ends of the cell, and spindle fibers fan out from them. Next, in metaphase 1, the homologous chromosomes line up at the equator and attach to spindle fibers from opposite poles. During anaphase 1, spindle fibers separate the homologous chromosomes in each tetrad and pull them to opposite poles of the cell. The cell enters telophase 1 with 1 chromosome from each homologous pair at separate poles. However, each chromosome still consists of sister chromatids. Keep in mind that each chromosome's sister chromatids are no longer identical because of the allele exchange that happened during crossing over. Then, spindle fibers disappear, and the nuclear membrane reforms around the chromosomes. Finally, cytokinesis occurs. Meiosis 1 ends with 2 genetically different haploid daughter cells. Each haploid cell contains only 1 set of chromosomes consisting of paired sister chromatids. Both cells now enter the next stage, meiosis 2. However, unlike meiosis 1, DNA does not replicate before meiosis 2 begins. Once again, in prophase 2, the nuclear membrane disappears, and spindle fibers fan out from the 2 sets of paired centrioles. During metaphase 2, the chromosomes in each cell line up at the equator and attach to spindle fibers from both poles. During anaphase 2, the sister chromatids of each chromosome separate and move to opposite poles. Once the sister chromatids separate, they're called chromosomes. Finally, during telophase 2, the spindle fibers disappear and nuclear membranes reform. And cytokinesis occurs in both cells. Meiosis 2 ends with 4 genetically different haploid daughter cells, each containing only 1 set of chromosomes. Some key points to remember about meiosis. It begins with a diploid cell. Meiosis only produces gametes. Gametes are genetically different haploid cells, sperm cells in males and eggs in females. Meiosis has 2 stages of cell division, called meiosis 1 and meiosis 2. During meiosis 1, homologous chromosomes separate to produce 2 haploid cells, each containing chromosomes in the form of paired sister chromatids. In meiosis 2, the sister chromatids separate in both cells, becoming individual chromosomes. Cytokinesis of these cells produces 4 genetically different haploid gametes. And here are some key points to remember about prophase 1. The pairing of homologous chromosomes called synapsis occurs. Each pair of homologous chromosomes, consisting of 4 chromatids, is called a tetrad. During the process of crossing over, chromosomes in homologous pairs exchange segments of alleles. Crossing over results in genetic differences in gametes. All gametes produced by meiosis are haploid.
Okay, hopefully now you have the general basis as to what's going on in folliculogenesis, which is the follicle development in the ovary, and meiosis in the ovary. And so yeah, let's continue. So this black image right here I found in her canvas, in the Lumio. This is what I'm assuming that she'll be testing us on during the final exam. And so yeah, let's, well, I'll break it down for you guys. The cell is 2N, which contains 46 chromosomes. So before I begin, I want to go over some terms. Diploid means 2N, haploid means 1N. And in here, it says that 1N is equal to 23 chromosomes. So therefore, if it's a diploid, it has 46 chromosomes. And if it's a haploid, it only has 23 chromosomes. So in metaphase 1, it has 46 chromosomes because it has 2 N's, right? So in here, it has 46 chromosomes. In, what's it called? In metaphase 1, what happens is, the homologous pairs line up each other on the metaphase plate, and in this phase, it contains the primary oocyte, So that's metaphase 1. In anaphase 1, moving here, the homologous pairs separate. So they separate, and if you recall, this is one end, this is another end. So what happens is the ends split. So now each end has their own cell now. So 23 chromosomes go to each cell. Therefore, they have one N, and therefore they are known as a haploid. And in the telophase 1, they contain the secondary oocyte. And that should be it with the first meiosis. Okay, moving on to meiosis 2. It looks like we have to know prophase 2, metaphase 2, and anaphase 2, but I'll summarize them in an easy way. So prophase 2, it looks like nothing's written, so I don't think nothing's really going on. Even if you did watch the video, it says like, oh, you know, during prophase 2, nothing really happens. Nothing exciting happens. And so yeah, prophase 2, we can skip it because nothing really happens. Moving on to metaphase 2. In metaphase 2, the sister chromatids line up. So the sister chromatids is this, right? What happens is they line up to separate in metaphase 2. And then in anaphase 2, the sister chromatids separate, therefore giving us 4 haploid daughter cells. And let's see. Yeah, that should be it with basically everything. All right. Now let's move on to the males. Now, thankfully for the males, we don't have to know that much because we don't have to know the lecture portion of it. She doesn't go over it in the lecture, so it's not going to be an exam. So going over some of the lab lists that we have, it's epididymis and testes. If you recall, this whole structure right here is the testes, and then sitting above the testes is the epididymis. So the first thing in our lab list is the seminiferous tubules. So what that is the structure inside the testes. Okay. Now looking over the seminiferous tubules in a histological sense, this is what it would look like. Okay. Now if you were to zoom in a little bit deeper, this is what it would look like. So this whole structure is still the seminiferous tubules. And then right here, you see that outer membrane? If you see a cell that's pointed inside there, that is called the interstitial cells. So like these cells right here. And then this right here is the primary spermatocytes. Jesus. You know, giving you a clearer picture, this is what the primary spermatocyte is, and then this is the secondary. Notice the secondary, it's smaller than the primary. Unlike, you know, the females where the primary, it's getting bigger as the stages go through, right? And that should be it with the male parts.